let's talk about the program. I am the moderator for today. I welcome you all to this topic today of the webinar, Systematic Monitoring Data. And I will start 10 minutes now giving you a quick overview over the field. And then we will start with the first presentation by Lisa Sundquist and Malin Werner on new data formats for marine ecological research. This will be followed by a presentation by Anna-Lena Axelsson on terrestrial vegetation monitoring data. And follow that, after a short break, we will continue with a presentation of data from birds and butterflies and mammals. And then we have one more on the BioCollect system by Alexandra. And we plan to finish the session today with a Q&A part at the end of the meeting. So we will not be working beyond 12 o'clock. We may finish earlier. Let's see how many questions we have. And short points on how we're planning to do this. The meeting, as you see already, is recorded, and we're planning to publish it on the website by the latest by the end of next week. So you can always go back and watch it or send it to a colleague to watch it. We ask you to stay all muted throughout the meeting. And if you have questions, please post them in the chat or write them down yourself so you can ask them at the end of the presentations. We're planning to take in a few questions, one or a few questions after each presentations, and I will try to pick them. Otherwise, we take the remaining questions and the more general questions at the end of the meeting. So please think about future developments and how you could add to this by suggesting certain types of data, certain monitoring programs you either would like to add to our infrastructure or to utilize yourself. And we really appreciate any kind of feedback and suggestions on that topic. And please use the support web form to come in contact with us. So here I show it like the same as I showed on Monday. There is always a support button on any of the pages that you're browsing. And when you enter this, when you press this button, you get to the support communication. And the important thing is that it gets to more than one person, so it's not getting lost in case somebody is on holiday or on sick leave. And with that, I think I've said everything about the introduction. So I would like to start with a short overview over what systematic monitoring data is. We consider it as a goldmine for researchers, and I also think that systematic monitoring data is really essential data for data-driven biodiversity research that we are trying to stimulate and promote in this infrastructure. Now, what does it mean to collect systematic monitoring data? There is, if you compare that to classical data that are collected by researchers, there is quite some differences here. The main features that describe systematic monitoring data is that it's standardized collection of data using fixed protocols. So it's very important that in these programs, the protocols are used and used again. And another important feature here is that it's repeated, it's reoccurring year by year by year, and that it's not necessarily related to a single hypothesis. That these are very important characteristics, how to distinguish systematic monitoring data from classical research data. Now, when you're looking at the history of systematic monitoring programs and the data coming from these programs, there has been a big change in the last 20 years. Until for a long time, these kind of data were considered and compared to kind of stamp collecting activities that were not related to any rigid hypothesis testing, and as a consequence, they were not really seen as a potentially valuable to use for scientific activities and scientific research. And these kind of dark ages for monitoring have now brightened up and became completely changed through the rise of research related to global change. So global change really provided a very good context to make these data so valuable. And most of it, of course, because of the effects of climate change that need to be investigated now by the research community. And if we want to understand climate change and the change of habitats and the effects that these changes have on biodiversity, we do need long-term time series. So thanks God that these programs were pursued and maintained throughout the last, let's say, 3 to 4 decades. And some of them managed to maintain their time series for such a long time. So here you see some of the papers that have come out, and many of you are already working with these papers. There is many hundreds of papers now published today based on systematic monitoring data, which we're trying now to make accessible through the infrastructure. And here you see some examples. Well, one thing that changed was in the scientific community, the archaeological community, is about 12 years ago that these data were considered to be really valuable, and there was a very good scientific discussion that how these data should be used and enter the scientific community, so to speak. So that scientific discussion basically led to the data-driven research that is now, in its essence, basically having the process has changed. You start first collecting data, and then you ask your hypothesis, which was kind of not so accepted before. Now it is a much more accepted and respected way of proceeding with research. How did that work in Sweden? Well, if we're looking at the history, here are the major institutions that have had responsibility to collect long-term monitoring data in the past. One is Naturvårdsverket, that's the Swedish environmental agency. The other one is the Länsstyrelserna, that is the county administrations or the county administrative boards. And then you have the Havs- och vattenmyndigheten, which is the Swedish Agency for Marine and Water Management. And associated to this, we also have the SMHI, Swedish Meteorological and Hydrological Institute. And these institutions have had usually the main responsibilities to collect data with systematic monitoring programs. So these are the data we are now trying to make accessible through SBDI. And this is only for Sweden. So we know that many of these processes and activities are happening in other countries as well. And there are European infrastructures that are growing now and trying to connect and link and make accessible these data on a larger scale. So we're connecting to these as well to make sure that the data for monitoring programs that we publish are kind of linked to the same data in other countries. Okay, what can you do with these data once you have access to them? Well, the magic word here is scale. You can now access data to look at biodiversity, trends, patterns, and processes over entire new scales. And here you see the amount of dead wood in Swedish forests. That illustration would have been impossible to create from data collected by a single scientific projects. So that is the big difference. If you have monitoring programs, you can investigate spatial scale, but also temporal scale. So here you see examples from bird trends for 2 birds from the Swedish bird survey, the Svensk Vogeltaxering, and you can investigate and compare all kinds of trends between species and groups. Now, that's for the land. We are very eager to collect the same data, similar data from the ocean, anything from plankton to seals. And here we have 3 large providers of data that we are making data available within SBDI. And here you see one example study we did like 5 years ago, where we really could investigate the entire Swedish west coast over a scale of 80 years. Let's say we could go back 80 years in time and compare the richness on the seafloor fauna. And you see on the right hand graph that there is an incredible drop of biodiversity on the seafloor that the fauna experienced in the last 80 years. It was almost minimized by half of the species. Now, that is only looking at 2 points in time, because the time series usually don't go far enough back. They're not so detailed beyond 30 years back in time. But if you take other data that SMHI has collected, you can see actually an opposite trend, at least for the West Coast. You see these red points. This is data from the West Coast collected from SMHI monitoring programs. And you see that the diversity actually has been rising slowly again since the 90s. So depending on what data you have, you can look at different time scales and see processes. And there is actually a positive effect coming based on these regulations that were starting to protect the ocean in the early 90s. Now, how are these trend lines helping us to do science and manage our environment? Well, there is a good comparison in terms of the natural capital that we are trying to investigate. So the role model here would be something like the stock market, where you are investigating the trends in economic capital. And the economic entities that you would investigate on the stock market is a company. While for us in the ecological world, it would be a species or a habitat. So there's all kind of biological entities that you can define, and then you can create these trend lines if you have access to the data. And if you continue that, you can also sum and average these trends across larger groups. You could say you group taxonomic entities into larger groups, such as butterflies or brittle stars. You could define the trends for larger groups, such as ecological entities, for example, pollinators or bioturbators. You can find all kinds of other indices to describe the trends for countries, for certain groups of species, like red-listed species, rare species, or non-indigenous species. And once we are able to map and monitor the trends of these ecological entities, then we are, just like on the stock market, able to foresee we have a certain amount of predictability, and we also have a certain ability to react to certain trends. And that is something where we are trying to work here in the medium and long term, to provide these data to understand, to monitor trends and to understand them, the causes of these trends, and then mitigate these. And with that, I want to stop here with my short plea for systematic monitoring data. And I will give the floor now to our first speaker for today. That is Lisa Lundqvist.
Yes, and I will start sharing my screen. Okay, can you see the presentation?
Yes.
Yeah, good. Thank you very much, Matthias, for that nice introduction. My name is Lisa Sundqvist, and I work as a data manager and a project leader at SMHI. I work with marine environmental monitoring data, primarily with biological data. And I have been part of the SBDI and former LifeWatch work since I started at SMHI nearly 5 years ago. So I'm very happy that we now finally are launching this new infrastructure. And I will start with some background. SMHI have more than 100 years of experience of conducting marine environmental monitoring and handling the data it produces. So we collect, manage, and present data. And SMHI does monthly cruises from the West Coast to the Baltic, now using the new research vessel Svea. And on these cruises, we collect physical, chemical, and plankton data from the open sea. We are also national data hosts for oceanographic and marine biological data, not including fish data, that is SLU, and Malin will tell you more about that later. We are appointed data host by the Swedish Agency for Marine and Water Management, and we archive, publish marine physical, chemical, and biological data collected within Swedish environmental monitoring program. And this means that we, as a data host, handle much more data than the one we collect ourselves. We are also, since 2017, appointed National Oceanographic Data Center, NODC, by UNESCO's IOC and IOD. And our database, as I think some of you know, is called SHARK, which is an acronym for Svenskt havsarkiv, Swedish Ocean Archive. And to get access to our data, we have Shark Web and Shark Data, where Shark Web is for manual filtering and downloading of the data, and Shark Data is an API, but it also has a simple web page for package downloading. And to give you an idea of the amount of data, at the moment we have about 1.3 million samples from 50,000 sampling points, 290,000 sampling events, and 7.3 million physical and chemical measurements and 4.4 million biological measurements. And before I go into details about export formats and file structures, I wanted to start talking a bit about Swedish environmental monitoring. And as Matthias mentioned in the introduction, Sweden have a long history of systematic environmental monitoring. And then now, when understanding the effects of global change is a top priority, we should be very thankful that these monitoring programs was initiated already a long time ago, because many of the Swedish monitoring time series are now starting to get quite long, and thus they are also very useful to study change over time. The Swedish Environmental Protection Agency, SIPA, and the Swedish Agency for Marine and Water Management, SWAM, also Hav- och Naturvårdsverket, are responsible for the Swedish environmental monitoring. And they are also responsible for the method descriptions, the so-called undersökningstyper, or recently actually renamed to övervakningsmanualer in Swedish. And this is the key to systematic monitoring, that all data are collected using the same methods, even if it's collected by different institutes and over many, many years. And in our data hostship, we have data collected according to 19 different methodologies, and of those, 17 are for biological data. And we have the following. We have bacterioplankton, chlorophyll, epibenthos, that's mostly macroalgae, but also macrofauna, phytoplankton, picoplankton, harbor seals, gray seals, ringed seals, harbor porpoise, seal pathology, sedimentation, zoobenthos, zooplankton, primary production, and physical and chemical data. And as you can see, the longest time series here are physical and chemical data, where we have measurements already from 1893. Unfortunately, the biological time series are not that long. Most of the programs started during the 70s, 80s, and 90s. The Swedish system with data hosts are important for the quality and not least the standardization of the data. And SIPA and Swarm appoint specific authorities and universities to be data hosts. And thus data are gathered in a few places close to the expertise. And even if it sometimes feels that we have a very long way left to go before we can say that the Swedish monitoring data is standardized and interoperable, working in international data projects, it is often my experience that Sweden have a very good accessibility and management of our data compared to many other countries. And I think the data hostship system is one reason for that. So data is received and maintained and stored and made available by the data hosts. And most data hosts have both a web page and an API for data access. And for a long time, the focus have been on gathering and publishing data so that all data should be freely available for everyone. And even if standardization is in the core of monitoring itself, the standardization of the data and the metadata is not as good as one could wish for, maybe. But during the last years, standardization between different data hosts has started to get more prioritized. And now the discussions are more about increasing quality controls, FAIR principle, making data not only accessible, but also interoperable to increase machine readability. But this is also something that increases the human readability a lot. And data hosts are working on standardizing data formats, even if many of the projects have shown to not be as easy tasks as, and it's now clear that it's very difficult to standardize in retrospect. For instance, we have worked on a national station register for I think it's over 10 years now. And it's still not totally clear how to define a monitoring station. And this is after hours and hours of meetings. And projects on standardization, standardizing formats, codes, and terms are also initiated, but also working quite slowly. And here I think it's important to not only see the Swedish environmental monitoring data as a Swedish resource, and instead have an international perspective. And here I also think that SBDI is very important since the data hosts are set to have a national focus. But SBDI can bring in a more international and more research-focused perspective on the data. And with the experience of how difficult it is to standardize in retrospect, even within Sweden, I think it is important to take international standards into account from the beginning when possible. Because this is what it looks like, at least for us. And I guess it's the same for many of the other data hosts, that having access to a lot of data also leads to projects outside of the data hostship, both nationally and internationally. And in this context, it starts to get interesting to talk about formats, because standardized formats makes it so much easier, both as a data provider and a data receiver. And for biological data, we have had the Darwin Core format for quite a long time, and we have delivered occurrences in Darwin Core for many years, both within the LifeWatch project to the analytic portal and to the EU project EMONET Biology. But now we also have the event core and the extended measurements of fact to connect to the occurrences. And this is, in my opinion, a huge step forward, because it's only so much you can do with occurrence data, and we have so much more information available connected to our occurrences. And in this picture, you can see an overview of the extended Darwin Core format that I took from the OBIS web page. And as you can see, you can fit quite a lot of information into this format. This is only one example. You can also use it, for example, for transect data. And here you can see that you can organize it by cruise or by station or by sample. And you can take subsamples where you can measure nitrate or grain size, for example. And you could have many occurrences, and to every occurrence you can link biomass, abundance, body length, for example. And the very cool thing is that it makes it possible to download rich biodiversity data, including a lot of measurements and metadata from different organism groups and from different areas of the world, and analyze it together in the same format. And of course, it would be naive to think that analysis could be made straightforward. We will still have quite a lot of work ahead of us to make the data standardized, quality assured, and interoperable. I have been involved in some work with an eMonnet to make data products. And it's not that easy to work with data from different countries because you have to put quite a lot of effort into making the data homogeneous and to know that you actually have the same thing. But I really believe that the work that we do now will increase the usability of the treasure that is Swedish environmental monitoring data, both nationally and internationally. And I really hope that extended Darwin Core format will make it easier for researchers to include more data in their analysis in the future. And we have worked intensively with putting our data into the extended Darwin Core format this year. And we managed to get most of our information in here for the exports that we have prepared so far. But we are putting quite a lot of information into dynamic properties, meaning that the information does not fit into any of the standardized Darwin Core terms. But hopefully the number of available terms will increase in the future. And I actually really like that the format is both standardized and flexible at the same time. It makes it very easy to use. And we have worked on trying to make as an as automated Darwin Core generator as possible, so that we could update it easy when we change things in the database. And I won't go into details about the program here, but it's available on GitHub, and anyone who is interested in checking it out and or have any questions can, of course, are very welcome to do so and to contact us if you have any questions or suggestions. The generator is written in Python, and the information that needs to be changed when adjusting exports are saved into YAML files that can easily be edited on your computer or directly on GitHub. And I'm actually very happy with the structure that we have now. We started out using big Excel spreadsheets, trying to map all our columns to the Darwin Core terms, and it was not that easy to get a good overview. But this system works very well for us, at least, and it's easy to get the overview of what you're mapping. And here is a slide of our data divided into different packages, where the dark green packages are the ones we have delivered to GBIF and SBDI so far. And the light green ones are data we hopefully can deliver in the future. And the light blue ones are biological data that do not have species observations, so those cannot be put into the Darwin Core format. When delivering data, we have divided the packages into national monitoring and other kinds of data. And that is regional data, regional monitoring, recipient control monitoring, and project data. And the reason for this is that we have a better control of the methodology and the quality of the national data. And we have very good regional data as well, but we don't really have a good system to separate the ones with good quality from the ones with less good quality. So at least for now, we have done this separation between national and other kinds of data. And this is the packages we have delivered to GBIF so far. They are generated through our generator, and then we just upload them at the IPT. And the mapping and the metadata and everything is there from the beginning, so it's just to publish. So that's very easy. And when you here is you can see what the data looks like in the SBDI portal. And And you get this very nice overview of the data set. And this description is a very good way to communicate with the user. And this is something that I sometimes miss in our own database, that we do not have a good way to in text tell someone something general about the data or the monitoring program. And I also think that this can be very useful if we use this text when we update the data. So for instance, if we 1 year have worked very much with the stations or done specific quality controls, that can be written here so that the user can clearly see what changes has been done. And at the portal, you also get this nice overview where you can see the distribution of the data. Here we have a phytoplankton data set, and you can see that we have data from 1983 and that the amount of data increased over the years. And we have data from all around the year, but most from spring and summer. And you can also see if you have any quality issues. Here, for instance, you have some names that are not recognized. And in this case, I think this is flagellates and unicells that are not determined. And you can also get an overview of the taxonomy of the data set. And then I just want to briefly show you what you get if you download the data. I won't go into details here, but you get these 5 files. And here you have just a small part of the event table. It contains a lot more columns than this, but here you get information like sample size, value, and event date, station, and plankton sampling method, and sampler type. And then we have the extended measurement of fact, where we have 7 taxa, and you have abundances and biovolume concentration for those. And then we also have the occurrence table, where you find information about the species that are linked to dune taxa and worms. And here you see one of these flagellates that are undetermined, and then we lack the links here. But this is just a small part of the data that contains much more than this. And if you were there on Tuesday, you already know all about this SBDI 4R work package. And if you missed that workshop, I think you can see it. It will be put on the webpage later. So we have, if you want to analyze the data, this package is available. I have not had time to work with this myself yet, but as I have understand it, the R package has not that much functionality available going outside of the occurrence records yet. But I really hope that this is something that we can develop in a near future. And that was all I had. So then I leave the floor to Malin, who will tell you a bit about SLU.
Yeah, my name is Malin Werner. I work at the Department of Aquatic Resources at SLU, we're shortly called SLU Aqua. And I just want to take the opportunity with this last slide in this presentation to tell you that there's also data on fish in SBDI. And we have done, the Department of Aquatic Resources, and before that, the Swedish Board of Fisheries have done systematic monitoring of fish for at least 50 years. But the data from there is in the sea is delivered to an international database. So for the moment, there is data on freshwater and coastal monitoring fish in SBDI, but the international surveys are mostly in the ICES website, but will be available in the SBDI portal too soon. And I wanted to add this that Lisa said about the event data, because when we do this monitoring, we actually are mostly focused on, as fish is a commercial product, we're mostly focusing on using the data for stock assessment. So we do a lot of other measurements like age and length and weight. So this way to also be able to show all these things in the extended format is really good for us. And the marine fish stocks, they are shared between different countries. So this actually started as international cooperation with this monitoring in the sea. So, and we will continue with that, but it's really nice to be able to also show this data in the Swedish context. So I just wanted to say that there's a lot of fish data in there, but there's more coming soon. Yeah, I didn't want to add much more there.
Thank you very much, Lisa.
Can I stop sharing?
Okay, thanks so much. So if there are any urgent questions from the audience, we can take one while I'm preparing this next presentation. Okay, I'm just getting ready. Okay, I'm just preparing the next presentation now. Okay, that is by. Okay, there is one question from Frederick Ronquist. Data like production and chlorophyll can be shared as map layers in SBDI. Any plans to do so?
Yeah, that would be a good idea. We are not working at that right now, but it's definitely something we can discuss to do in the future.
Good. And then we are ready for the next presentation by Anna-Lena Axelsson.
Yeah, my name is Anna-Lena Axelsson, and I work at SLU at the forest faculty, and I worked with data from the Swedish NFI for quite a long time.
Anna-Lena?
Yes.
You're showing the version of.
Oh, sorry. I will.
If you have some.
Swap it now. You see the right one. That's
yes.
right. Yeah,
yes.
good. I work with open data from these monitoring programs for a long time, and especially from the Swedish forest inventory, national forest inventory. But for some years ago, we decided to put all the national terrestrial inventories together in a family. Åke is sometimes talking about a family of systematic monitoring programs, but we have a small family at SLU also where we have the terrestrial focus. And today I will present what we have, some basics about these inventories, so you understand that they're quite different. We have the National Forest Inventory that's been going on since the 1920s. And we have the Forest Soil Inventory, which is performed on some of the plots of the Swedish NFI. You can say it started in 1983, but there are also older data available. Then we have some raster products that are made from the National Forest Inventory data, which is another way of looking at the data. You have wall-to-wall maps. And then we also have some smaller inventories that are collected under umbrella, the National Inventories of Landscapes Sweden. And I will start with the Swedish NFI because that's where we have most of the data, and we have the long time series going back to the 1923. But you can say that the most valuable data for biodiversity research was collected from 1983. And what we will work with mostly with the SBDI is to provide ground vegetation data that are collected on some of the permanent sample plots. And there we have presence absence data, and we have which is collected from a coverage list or from a species list. And it's common species, of course. It's not rare species. So from a biodiversity perspective, it's maybe some people are interested in common species and some people are interested in very rare species. But we cater for common species. With this inventory, it's not possible to capture information on rare species. And we will fill in data every year. So we have a yearly delivery to the SBDI. And right now the data is at Artdatabanken, which will work on a connection to GBIF and SBDI. So the data are there, and I don't know really when we will have the data up at SBD portal. But we have made the first delivery, and soon we will add more data because our statistics was published last week, and then we can also provide new data for research. That's where we open up the data, which have been controlled, of course, thoroughly. So the data is really good quality because it's used for doing the national statistics. So of course you can always find problems, but we do a lot of washing and checking of this data because it's used to make the official statistics. And we also have these trends, not for the species though. Of course we can have trends for terrestrial vegetation from 1983 and onwards. But we have also, of course, a lot of data on the habitat, which is, of course, of real interest for biodiversity research. But the SBDI is oriented towards species observation. It's not that easy to provide these trends and these maps into the SBDI portal, and that's something that I've been thinking of a lot. And I think we have to work on this in some way. But I also think that researchers maybe use other tools and want to do analysis outside of the SBDI. So for us, the SBDI portal is mainly a window where we can show a showing window where we can show some of our data, and then we can attract people to us and help them with more data, because we will not be able to get all our data into the portal. And then we have also, just to show you what we're doing, the presence and absence vegetation is performed on one plot size, and then we collect other stuff, other information on other plot sizes. So it's quite a complex database structure, which is not easy to get into a flat file that easy. You can, of course, but you lose a lot of information and about the structure of the data, which is very important for when you want to do analysis. Here's another way of showing what we collect. So the information can be used for a great variety of things, but of course, for biodiversity, it's mainly the habitat information that is useful. And we have statistics, open statistics and services for APIs where you can collect the data or the statistics, the aggregated statistics at regional level. And I think these long-term trends could be useful for some research also, depending on your question. You can have really good use of these long-term trends. And we have recently added also plots in the Alpine region. So now we have data for all of Sweden, all vegetation. We capture information on all vegetation in a systematic way in Sweden. And we didn't do a lot of inventory in the Alpine birch forest before. mainly because the inventory was earlier aiming at the production aspect of the forest, but recently more environmental aspects are being included in the inventory. So that's why we look at all the vegetation. And the data is used in many different projects, of course, and for reporting, and also for doing forecasting. We have tools for doing forecasting also, and that could also be used for biodiversity aspects of the forest, for habitat, forecasting habitats. And as I told you before, the soil survey is performed on some of the NFI plots, and there we have a really nice inventory of DNA of soil organisms and also metabarcoding of these. And this is a really, really interesting inventory. And here you have all the information on the plots. You have information on the forest, and you can connect that to the soil organisms and to explain why they are there. And that's a really, really strong and interesting data set. And we have a portal here for that. And in the future, this data will be in some way, we will get this data into GBIF and SBDI, but we will not get the habitat information there. Maybe an ID for the plot where this mushroom or its fungi has been found, and then you can connect back if you want the information on the plot. But of course, if there is this large interest through this channel, we could put effort into this, but we will try and see first to see how much funding and how much energy we should put into this, providing information through this channel, because we provide data through other channels also. SBDI and GBIF is one of our channels where we send data. And then we have this landscape inventories that has been shifting a lot over time. Maybe some of you know about the NILS program. NILS is now an umbrella for many different regional programs. You have the basic NILS inventory, you have the inventory of seashores, and you have the inventory of grasslands. And the new inventory is dealing with deciduous forest. And then there was this inventory of the mountain area before, which is finished. So NILS is changing a lot. But they have their own portal, which is developed through this data delivery scheme with Naturvårdsverket, and through that they will provide data to GBIF and SBDI, and they have funding within this work that they do already. So the delivery to GBIF won't be a really big thing. They will just do another formatting of the data and send it through this channel. And the portal is preliminary, but if you contact me, you could get access to some of the data. I think the mountain area data is out. And then we have these raster maps, which is really, really useful if you want to do modeling. If you want to model biodiversity in a regional or national level. So you have a Zulu forest map, a basic forest variables, which has been used by a lot of ecologists for landscape modeling. And we also have a new map, the wet area map, which also is useful for research because, as you know, wetness index is a really important part of terrestrial vegetation. So we think that this map could be really useful for landscape ecologists who want to do modeling. And these maps are available through the GET service, which all Swedish universities have access to. And the maps are also displayed in the map services at the Swedish Forest Agency. So we cooperate a lot with them concerning these geodata part, and they have a very strong work, and they do really good work with this. So researchers that are interested to use this, they can go to the Swedish Forest Agency to look at the data, and if they're interested to use it in their own GIS, they should download the data. And we also discuss if we should get these services into the SBDI. But we have to discuss this more, because I think most of the people, the researchers that are doing landscape, do work with landscapes, they work with GIS. They prefer to work with their own tools. But there could be an aspect of that you could be able to look at a map as a background map. That could be maybe possible to do. We will discuss this further, how we could proceed with this, with these map layers. As I've discussed with you before, we have a complex data structure, and therefore we have decided not to put all the data into SBDI. So instead we provide tailored data sets that are aiming fits specific user groups or research questions. And one example is the ground vegetation coverage data, which is at Databanken right now and will be soon included in the SBDI. And then maybe we could also include trend lines, if that is of interest to researchers, the statistics. But then we have to find a way to do that technically into the SBDI portal. That's something we could discuss also if that is of interest. And here is another way of how we do it. We instead of putting everything out as open data, we also provide free help to people, to researchers that wants to use our data. And if you have a simple question, we provide sample plots data or raw data to the researchers. If this is a more complex question, they have to pay, but it's not that much money actually, and you get a really good data set that is really adapted to your research question. So that's some of the funding that we have in the SBDI is actually used to helping researchers to find out the right delivery for their purposes. So that's an important part of our work in the SBDI. And we have some restrictions. We cannot leave out coordinates of the permanent plots, but researchers can apply to get access to that. We have softened up the policy a bit. That's really good. And we have two models for data consultation. You can pay for the time spent to extract data, or you can also invite our analysts as co-authors in research cooperation, and then there will probably be no cost for getting the data from us. And it's interesting to see that more and more global studies are using our data. These are two recent publications where there are consortia, global consortia, which are collecting data from many different countries and working with global questions. And our data is really useful for doing this, as there are also other similar inventories in other countries. Just finish with saying that we have a lab which we do together with the Data Lab, which we work with together with the Swedish Forest Agency, and where we work to different user groups, and researchers are one of the user groups where we're aiming at. So far, most of the activities have been in Swedish, but we will planning to have more English events. I stop there. Thank you very much.
Thank you so much, Anna-Lena. And now, unfortunately, we won't have time to take any questions right away because we are a little behind in schedule. So let's have a break until 5 minutes past 11, and then we see all each other. See you in 10 minutes. Okay. Now it's 11:05 and we continue now with our next presentation by Anneli Jönsson from Lund University. She will tell us more on birds, butterflies, and mammals, many decades of data from systematic monitoring programs. So Anneli, welcome. You can take over the screen.
Thank you. There we go. Right, so thank you. I'm, like Matthias said, Anneli Jönsson, and I work at the Department of Biology at Lund University. I work with a data host project, just like Lisa was telling you about, and which was assigned to us by the Swedish Environmental Protection Agency. So we deal with data sets from systematic monitoring on birds and butterflies mainly. I will, in this presentation, I will tell you which data are available, or at least will be available from us, a bit about how they are produced, and then I will focus on how you can find them on SBDI, and I will also mention a few things about what it is you're actually getting when you're downloading data. And I will give this presentation to you as potential data users as well as publishers of data. That's my angle. So here's the full list of data sets that we are responsible for in our data host project. On the left here, we have two major schemes, the Swedish Bird Survey and Swedish Butterfly Monitoring Scheme. They're actually managed by my colleagues in-house, so to speak. Both schemes are financed by the Swedish Environmental Protection Agency, and all 21 county administrative boards they collaborate with and support these schemes. And the mission is, of course, to monitor population and distribution changes among Swedish birds and butterflies, and a bit more recently, also mammals. On the right, instead, we have some external data sets from survey programs that are managed mainly by the county administrative boards, länsstyrelserna. And I will use this in-house monitoring scheme to describe, as an example, to describe the kind of data that we have and why they are actually systematic. Let's point out, we have the oldest data set we have is the waterfowl count that actually starts in 1967. And the most recent program we have is the Swedish coastal bird monitoring that started in 2015. Anyhow, the fixed routes from the Swedish bird survey, the so-called standardrouterna. I actually chose this background picture to illustrate that sites are systematically placed without regard for the convenience of the surveyor. The view that you're seeing is the actual path facing a surveyor who's about to count birds along a transect on an actual site. In the program, there are 716 squares, and they are evenly distributed across Sweden. So this means that each habitat in Sweden is surveyed in proportion to how much there is of it in the country. Of course, all the sites are surveyed using a standardized method, a systematic method, which is, like Matthias said, repeated year after year after year. The sites are in this survey 2 by 2 kilometer squares. They are visited once a year in late May to early July. Each site consists of 8 1 kilometer transects and 8 point counts. Along all these transects and on all these points, all birds heard or seen are noted, regardless of how far away they are. But in general, they're more or less to plus minus 200 meters from the transect or the point. Some trivia from last year to show the scale of this scheme. We had 235 people walking about 4,400 kilometers while they observed 166,000 birds of 224 species. Almost all the species that we have in the database were observed. And data in this program stretches back from 1996, and the mammals were started in 2011. The data that is collected can obviously be used to calculate trends and distributions for these various species. The data can also be used for so-called miljömålsindikatorer. Not sure about the English word for that, but also for reporting to various EU directives, etc. But it's not only environmental monitoring we can use this data for, also basic and applied science, like also Matthias mentioned, the effects of climate change. So here we have a few examples of studies that have been done on these data. So basically the same data or different parts of these data can be used and reused to answer different questions. Here we have the full list of our data sets again, and the ones highlighted with yellow, they are the ones that we have published so far. So 3 of the data sets from the Swedish bird survey. We have published the Swedish butterfly monitoring scheme, and then 1 of the external data sets, the coastal birds in Gulf of Bothnia, kustfåglar i Bottniska viken. I will now spend the rest of the presentation to show you how you actually find these data on the SBDI webpage. So the first step is obviously to go to that webpage, biodiversitydata.se. On this page, you go to the Data and Tools tab. You click that, and you end up on this page. Here, under Swedish biodiversity data, you click on Datasets. That takes you to a page where all the data sets that are available on the SBDI are listed in alphabetical order. So you either scroll through or click through this list of data sets until you find what you're looking for, or you type in the whole name or part of the name here in this box and click search. As you can see, here we have the 4 data sets in our collection that start with the letter S. We have given the full English name of the program, but also included the Swedish name in the title, so you can search on both. Makes it easier to find them. In case you don't actually know the name of the data set you're looking for, but if you know who's providing it or publishing it, you can also find and get access to the data sets through this link. If you go back to this page and click Swedish data providers, then you get a list of data providers in alphabetical order, and under each will be all the data sets that they're responsible for. However, I stick to the this data set route when I continue, and I will explain why. So back to this list of our data sets, and we will look at one in particular to show you what it looks like. I've chosen standardrutorna again. So if you click on that name, the survey program's name, this is the page that you get to. Here's the top of the page, and here's the bottom of the page. I had to split it so I could fit it on the screen. But basically, you get the description that Lisa was talking about. You get some information about where the data is collected and why, something about the quality and methods, more about that later. And we have these lovely graphs that Lisa also explained to you. So this is the metadata that is provided. If you want to look at the actual data, you have 2 options. You have the button here at the top or the link further down the bottom. So if you click one of those, you get a long list of all the records that are in the data set. So of course we dive deeper and look at one record. I've chosen this one just randomly. This is what you see then. So this is all the data for this record that is in the data set, divided into sections for easier overview. So you have information about when it was actually recorded, you have information about the taxonomy, species, etc. And also, of course, where. There is a bit of information about data quality. Nothing major in this instance, luckily. There is just a warning to say that some data that is provided when you download the data has been sort of inferred or assumed. We didn't supply we didn't actually state in the data set that this species was present. But obviously, because we have a number of individuals observed, it has been assumed to be present, which is entirely correct. Once we looked at this, and you can see that the data is kind of what you're looking for, and it will be very useful for you in your analysis, then you go back to your the previous page, and you basically hit the download button, and the data is yours. But what is it then you're getting? What are you getting when you're downloading data? I will show you that, and I will also mention a few things to be aware of. Like Lisa told you and showed you, we also publish our data onto GBIF through the IPT as Darwin Core archives. So they consist of 3 different tables, CSV files, the event, the occurrence, and the EMOF, the extended measurements or facts files. And all these 3 files are connected or linked together through unique event and occurrence IDs. When you download data from SBDI, instead you get one Excel file, which is great in one way because you don't have to fiddle about with linking tables together with IDs. Unfortunately, the data that is in the EMOF will not be included. This is because GBIF doesn't yet support that extension, and they can't actually include it in the data that is published. Now, I was going to show you, well, Lisa already showed you what a Darwin Core archive looks like. So we have the 3 different tables, and this is supposed to be the filter, just to show you what kind of information could be in the EMOF, so information that you could be missing out on. This example is a survey program where it's, in some counties, it's mandatory to count either pulley, and in some places it's voluntary. So here we have included the information about whether the surveyor has counted them or not, so that the data user knows whether to expect this data or not. Likewise, the pulley, when they are observed, they should be ranked according to size. So that data is also put in the EMOF because it doesn't fit in the other tables. There is no Darwin Core term to that equals pulley size class. But there is a way around this. There is a way of getting this data, and that's if you actually go to GBIF. I know we're doing an SBDI webinar, so we shouldn't talk too much about GBIF, but I think it's important to know where you can get all the data. So if you pop over to GBIF quickly, then you go to a datasets page, you click the download button, and then you choose this alternative, the source archive. Then you get what Lisa showed you, the 5 different files, which includes the EMOF. You then have to link them all together yourself, but at least you got everything exactly how we publish it onto GBIF. Some other things that I've noticed when I've been comparing what's been published with what we actually uploaded. There are some fields that are missing from the event and occurrence tables. I don't know why yet, but I've used the support form to highlight this, and hopefully we'll get an answer. Also, both GBIF and SBDI, they run your data through some validation process in this publishing process. And for certain fields, there are specific code lists or vocabularies that are used. So if your values that you have used in this field, if they are not actually on that vocabulary, then you might end up with an empty cell. And but SBDI actually provides a table that gives you a comparison of the original versus the process data. So if when you're looking at one of those specific records, this is the same view that I showed before. So you've chosen one of the records in a data set. This is what you see. There is this button here, the compare original versus processed values. If you click that, you get this table where you have a column here with original values, as in how we actually supply it when we upload it through the IPT, and then a column to state what has been filled in the validation process. In this example, there was nothing major wrong or changed. The only thing I could spot was really that we supply scientific name as just Emberiza hortulana, and the process has filled in the author and the year and a scientific name. But otherwise, this was fine. And lastly, just a couple of words about metadata. When you download the data from SBDI or the recommended route on GBIF, you don't actually get any metadata in the download. If you choose a source archive, you do. The page I showed you earlier as well, the metadata on the web page, we go back to that as well. This metadata page here with a description and everything. At the moment, at least for our data sets, there is some missing metadata, like the method section. That is not properly filled in here. We have supplied a lot more information about methods, but it's, I guess there's a mapping problem or something for getting that information here. Luckily, there is a link to a GBIF metadata webpage. So if you click that link, then you will see all the metadata that has been supplied for this data set. And that was all I had for you. On that note, I say thank you very much. And if you want any more details, please ask or contact us. And I'll say if you want to ask anything about the data sets that we do have Åke Lindström with us as well, and he can help me answer questions about that. Thank you.
Thank you very much, Anneli. That was very nice presentation. I think we have room for one question. If anyone don't want to wait to the end, we can have one question for Anneli or Åke. Otherwise, we can continue now with the last presentation by Alexandra Makziarek. Alexandra, you can take the screen and she will introduce us to the BioCollect tool.
Hello, everyone. I'm Alexandra Makziarek. I am a programmer. Like Anneli, I work at Lund University in the Biodiversity department. But I work on a technical team with Johan Beckman and Mathieu Blanchet. In Anneli's presentation, you were introduced to the different schemes that we have at LU, and you heard about the data. I would like to tell you about the new channel that we have for the data to be submitted to us, which is a web application called BioCollect. I will tell you why we chose to work on this particular application, what functionality you can expect, and how it can be adapted for other systematic monitoring schemes. People usually make observations in the field for systematic monitoring schemes. They take notes on paper quite often, and at home they type up the observations into Excel spreadsheets. Then they send it via email to administrators of the schemes. Then the administrators check if there is any data missing or any suspicious records, and if everything is fine, then they enter records into a database. BioCollect is meant to cut into parts of this workflow to save the admins from performing some mundane tasks and to further minimize errors with data entry. BioCollect is a tool that allows researchers to set up projects with specified systematic inventory schemes and coordinate the work with staff or volunteers collecting data. The tool was developed in Australia as part of the Atlas of the Living Australia framework. Why we are working on this is because almost 2 years ago, a group of researchers at LU working on systematic monitoring schemes of birds and butterflies asked us to come up with a software solution to enable managing monitoring schemes and storing data. We didn't want to reinvent the wheel, so we didn't want to build a whole new application. So we researched a few existing options and we decided to go with BioCollect. There are multiple reasons why we decided to further develop this application. As LU had then been part of BioAtlas, which in turn was part of the Living Atlases community, it seemed natural to go for a component within this framework, and BioCollect is within the Living Atlases framework. Later, as we know, BioAtlas was developed into SBDI, and we wanted to be an integral part of this group. And BioCollect is integrated with other SBDI components like the spatial portal, species list, and the central authentication service where users can register. It's also open source, which meant that we could access the code and build on someone else's work, adding new features and improving existing ones. And we didn't have to pay any license fees. We're also through this part of a bigger whole where ideas can be exchanged and work can be freely shared between different organizations. And that hopefully contributes to the sustainability of the project. BioCollect is actively developed by a team at the Australian Research Council, and it's used by many projects in Australia. And we have already proudly contributed to the core code base of BioCollect by providing the internationalization feature and a record validation feature. And lastly, one of the factors for us was also that BioCollect uses modern technology that we wanted to work with. However, BioCollect wasn't an out-of-the-box solution for us for systematic monitoring schemes. It had to be adapted slightly. The projects that operate in Australia didn't have as strict methodology as systematic monitoring projects in Sweden do. For instance, it was used for citizen science projects mostly. So we had to add project administration tools to it so that I will talk about them later in more detail. And we also needed maximum control over the data entered and customized survey forms that enable validation of incoming data. Our schemes are based on repeated visits to complex geographical locations, like Annelie showed you. And these locations include a parent location and segments of points where the observations are conducted. So they are hierarchical. And we needed to develop that feature in the application. We also have predefined sites, which can be booked by volunteers, another admin feature that had to be added. We have two kinds of users for this app, but there are multiple roles that can be assigned. For us, the most important ones are the volunteers who provide the data and the admins who manage volunteers and control the incoming data. I will start with volunteers. The volunteers can create new sites where they want to conduct observations. These sites have to be confirmed by administrators. The volunteers can also display the site later, download the coordinates, or save a map to use a paper map in the field. They can also see which pre-existing sites are available, and they can request booking one or many. On the right, you see one of the main functionalities. The screenshot is really small, so you probably can't read it, but this is the layout of a form for one of the schemes. This is how the volunteers report observations, where they enter species data and some metadata for the event of when the survey was conducted. A lot of what we created in this category of volunteer functionality was meant to offload tasks from the administrators. So, for example, the volunteers can change their contact details themselves, and they submit the data directly into the database, which is then verified by the admins, rather than the admins having to deal with paper surveys or Excel spreadsheets submitted. The other user group is administrators of projects. They can do everything that the volunteers can do, plus they have some enhanced functionality. They can edit and retrieve volunteers' contact details, and they get notifications about the volunteers' activity. They can see an audit of changes to survey forms. They can also create new projects, manage surveys where they determine what kind of species can be entered for the species lists in SBDI. They can also book sites for volunteers, validate both sites and records, and to deal with volunteers who still don't want to register online and use the application, we also have functionality where they can upload data from spreadsheets submitted via email. Through the work that we have already done, extending the tool for systematic monitoring projects, we are hoping that other systematic monitoring schemes can benefit from our work and set up their own instances of the tool. BioCollect was built with the idea that it could be adapted for other projects by creating a page using BioCollect's graphical user interface, like in a content management system, like WordPress, for example, where you don't have to be a programmer to create a new page. However, how things are now, if you would like to set up your own instance, that's the technical work that has to be done. The application has to be deployed and the database has to be deployed on a server. And unfortunately, we can't do that for other projects, but we can provide support and explain the whole procedure. And the code for it is out there and it's for free. And in the screenshots before, you saw pages from Svensk Fågeltaxerings, the bird monitoring schemes branding, but without it can be all adapted to the branding of the project that wants to use the tool. And that's it from me. Thank you for your attention. Again, the technical team that I work on is Johan Beckman, Mathieu Blanchet, and me, Alexandra Magzarek. You can contact us through this email address, biocollect@biodiversitydata.se. And if you'd like to see the tool, you can also access it through the link, biocollect@biodiversitydata.se. However, you have to register. So if you are a bird watcher, then you can just do it yourself through SBDI. But if you'd just like to test the tools for some other systematic monitoring schemes, please contact us and we can arrange a test account for you so that you can see how it works. Thank you very much.
Thank you so much, Alexandra. And I think we now arrived at the end of our demonstrations and presentations, so we can open the floor for discussions and questions if there are any. We see there were already 3 questions in the chat. They were in response during the talk by Anna-Lena. Now, Anna-Lena unfortunately is not able to be here, so that was a recording. She said she would look at the questions right away. I sent an email with them, but she hasn't come back to me yet. So I'm continuously monitoring my email to get the answers. But maybe any one of maybe any one of the other people joining today are able to answer the question. So Anna Lysch Solheim, she raised a hand. Maybe you want to rephrase your question, Anna.
Yes, thank you so much. I'm based in Norway at the Norwegian Institute for Water Research, and I work a lot with the biological data that are collected in the Water Framework Directive monitoring schemes. I also work a lot for the European Environment Agency and in the Topic Center for Water and Inland and Coastal and Marine Waters, collaborating with the Biodiversity Topic Center as well. I'm also a partner in the Europa Bond project, the Horizon 2020 project, aiming at creating harmonized design for biodiversity monitoring across Europe for both terrestrial and aquatic ecosystems. So this is just my background. I'm really impressed by what you have shown me today, and I've learned a lot. And I'm wondering if you can say a bit more about how you managed to do this. You build up a long time series by monitoring different sites, and you have shown several maps where you have these raster points covering all of Sweden. Does this mean that you actually do monitor you have field observations from all those sites, or do you use models to extrapolate from a few sites to cover all of Sweden? I'm really curious about that, because to have spatial representativity is essential in order to get a credible map of Europe. If your model for biodiversity monitoring can maybe be a model for also other countries, then I think it would be really nice to know a bit more about your way of extrapolating to maybe non-monitored areas.
Thank you. Thank you, Anne. Yeah, so maybe we should take that question to Åke and his group on the Swedish bird monitoring data. Do you extrapolate your data?
Thanks and hi. Yeah, where should I start? I think in terms, so we have made one collaborative study with the forest inventory, which was that's when you asked your question. And the data we get from them are sort of area. So they have extrapolated their small dots into larger areas with some kind of models. And then we compare abundance of birds in the corresponding area. So of course we are not monitoring everywhere. So I'm not sure how they sort of expand their data into for a raster or for a complete coverage. So Anna-Lena would need to answer that. What I can say with especially the fixed routes is that by having this systematic grid put out, we will, at least for all habitats of major distribution that are more common, we will really have a complete representative picture of what's happening at these sites. So what we measure there will be representative for larger areas. And then, of course, for every single site, like it's a single bird survey route, that could be by chance not very representative for that exact county or so. But the larger scale you use, the more representative they will be. And I can only say that if you're not aware of it, both Norway and Finland have adapted the idea of our fixed routes. So there are very comparable systems implemented in both Norway and Finland since 2006, I think, and we are already producing a lot of joint Nordic trends and distribution changes analysis and so on, based on the same idea of having systematic and representative sites.
Thank you, Åke.
Thank you so much.
Maybe you should quickly ask also Lisa, how much are the SMHI data from the systematic monitoring programs, how much are they extrapolated?
So they are not not in the. Yeah.
Yes, I haven't seen anything like it as well. It's the raw observations at the monitoring stations that you can use. But yeah, we have produced some models based on these data, so anyone can use them to create raster maps of species distributions or habitat distributions based on the data. Good. That was the first question. Then second question came by Tobias Anderman. Are only wood plants counted in the forest inventory plots, or are all vascular plants counted? Now, I'm not able to answer that, but coming back to that, what Åke said, have you received any of those data on any not wooden plants in the collaboration with Anna-Lena? Or was it only
we only used real like forest data, so nothing on what's on the ground. No.
Okay. Yeah, and then Judith Weber also asked maybe a general question: how good is data quality defined and how it is controlled manually, automatically? Well, I can, from what I know on the different data providers, there is both levels of quality control. That is very typically in all of these monitoring schemes, that there is a certain degree of automation in the quality control, but there is definitely also a lot of manual curation of the data sets happening. If any one of you want to add to this question.
I can say that it's a very good question, and whatever kind of test systems you have and control systems, bad things will always slip through. For example, what we do with the bird data is, in principle, we go through every observation and then write back to the observer. We have 800 people around Sweden, that volunteers that count the bird data. So we will actually go back to each of them via mail or phone and ask them, was this really so and so, etc. But we can still find errors from 1990 and 2011. That's certain when we do some special research on those data. So yeah, things will always slip through, unfortunately.
Thanks. Now, a question from Frederick Ronquist. In SBDI, we are planning to provide some particular valuable pre-computed data products. Various model-based extrapolations of point data to map layers might be interesting targets for these activities. Ideas on priorities are welcome. So that was the actual question. If there are any suggestions by scientists who would like to see any kind of data products in form of layers, data packages, then yeah, you just let us know. Good. And are they yeah, it's 11:45, so if there are no further questions, then I think we can close the webinar here. And I thank all the presenters for their very nice, informative, excellent contributions. And everybody else, many of the questions that were raised, we will add them to the frequently asked questions site on the SBDI webpage. And we will also make the webinars available from this week. And with that, okay, yes, there was one more from Anna. Yes, Anna says, data products for biological groups in rivers and lakes. based on water framework directive monitoring. We will definitely pick that up and may get back to you about this. Thank you, Anna, and thank you everybody for attending. And
thanks a lot. And good luck.
Okay.
With the further development.
Yes, yes. Thanks so much. And bye-bye, everybody.
Bye.
Bye-bye.